Welcome to this explainer. Today, we're going to completely dismantle your assumptions about the fundamental ingredients required for life to exist. For decades, our search for extraterrestrial life has been strictly governed by what we see in the mirror. But what if life doesn't actually need water? What if it doesn't need carbon at all? What if the building blocks of biology are vastly more diverse than our own little planet suggests? Let's get right into it.
So, to really wrap our heads around this, we first need to confront a concept known as our Terran bias. It's this limiting, though completely natural, assumption that all extraterrestrial life just has to rely on the exact same carbon and water architecture that we do here on Earth. I mean, think about it. If a sentient computer made entirely of silicon were out there scanning the galaxy for intelligence, it'd probably exclusively search for other silicon-based life, right? It would completely overlook us. To truly search the cosmos, we've got to broaden our horizons way beyond Earth's specific geochemical history.
Section 1. Beyond life as we know it.
If we're going to search for exotic life, we first have to agree on what life actually is. Take a look at NASA's working, operational definition of life. Notice what's missing? It's completely blind to chemical composition. There's literally no mention of carbon, no mention of water, and absolutely zero mention of DNA. It sets a really profound ground rule. Life is defined by its processes, things like self-replication and Darwinian evolution, not by its ingredients. This abstraction is incredibly powerful because it's our ultimate key to unlocking life as we don't know it.
Section 2. Carbon and water alternatives.
Okay, so if life is basically just an evolutionary process of interacting molecules, let's look at how we might swap out those fundamental building blocks. On Earth, carbon is the undisputed king. But there are other elements waiting in the wings. Silicon, which sits right below carbon on the periodic table, can form very similar molecular scaffolds. That means silicon-based life isn't just a trope for sci-fi villains. Boron can do the exact same thing. And it gets wilder. We could theoretically see life based on metal oxides and sulfur. In fact, laboratory experiments have already formed inorganic chemical cells called ice shells that mimic biological membranes using metals. It totally proves that complex, lifelike chemistry can happen in some incredibly unexpected ways.
And then there's water. We're always told to follow the water, right? But here's the crazy part. Water is actually highly destructive. It actively degrades and hydrolyzes many of the organic molecules needed for early life. Astrobiologists call this the water paradox. Because of this, alternative mediums might actually be vastly superior for maintaining stable, exotic biochemistries. Take formamide, for instance. It won't destroy essential organic compounds, and it stays liquid all the way up to 210 degrees Celsius. Or picture the freezing moon Titan, where massive lakes of liquid methane could perfectly support a completely bizarre, non-polar hydrocarbon biology.
Section 3. Harvesting alien energy sources.
All right, so we've got new building blocks and new liquids, but how do you actually power an alien organism in a pitch-black subsurface ocean millions of miles away from the sun? Enter the hypothetical mechanotroph. Instead of relying on photosynthesis, this kind of organism would act like a biological windmill. It uses the physical flow of ocean currents, or wind, to literally spin an external flagellum. This physical rotation cranks an internal motor protein to directly generate chemical energy, like ATP. It's literally harvesting the kinetic energy of its environment just to stay alive. How cool is that?
Equally bizarre is the osmotroph. Imagine a microbe just bobbing near the icy crust of Jupiter's moon Europa. Instead of eating or absorbing light, it generates energy simply by exploiting osmotic gradients. As water moves in and out of the cell across a membrane due to changing salt concentrations, the organism transduces that movement into high-energy covalent bonds. No sunlight, no standard food required, it's just riding the local salinity waves of its alien environment.
Section 4. Rewriting the genetic code.
Now let's move from theory into the lab. We're transitioning to the incredible science of xenobiology, where scientists aren't just looking for exotic life in space. They're literally building it right here on Earth. Compared to our natural biology, synthetic xenobiology is like upgrading from a flip phone to a quantum computer. Nature gave us DNA, RNA, and just 20 standard amino acids to build every single living thing you've ever seen. But xenobiologists, they're exploring a massive universe of over 700 alternative amino acids, and even XNAs, or XNAs, which use completely different chemical backbones. We are no longer chained to Earth's evolutionary history.
We're rapidly recoding life's operating system, and the pace is frankly staggering. For billions of years, life relied on just 4 standard letters, A, T, G, and C. But look at this acceleration. By the early 2000s, scientists were creating unnatural synthetic base pairs in the lab. In 2014,
Living bacteria actually replicated using artificial nucleotides inside them. And by 2019, Hachimoji DNA was born, an entirely synthetic 8-letter genetic system with massive information density. We literally doubled the genetic alphabet in a fraction of a human lifetime.
Section 5. Building alien life inside.
So, you might be wondering, how do you actually boot up this new genetic software in a living, breathing bacterial cell? They do it through an incredibly meticulous process called genetic code engineering. Researchers basically hack a bacterial genome, systematically replacing thousands of specific stop codons just to free up space in the genetic dictionary. Once liberated, scientists program those newly freed codes to force the cell to use completely non-natural amino acids. They even use directed evolution to make the organism entirely dependent on synthetic analogs, like 5-chlorouracil. What that means is the bacteria literally cannot survive without human intervention and a steady diet of synthetic chemicals.
Section 6. The ultimate genetic firewall.
Now, creating this parallel shadow biosphere in the lab isn't just a fun science experiment. It actually solves one of our absolute biggest biosafety hurdles. By making these organisms genetically incompatible with natural life, scientists construct a genetic firewall known as semantic containment. If a synthetic microbe escapes into the wild, it can't survive, and it certainly can't mix its DNA with wild organisms. But it gets even better. Because its genetic code is literally alien, it is completely immune to every natural virus and bacteriophage on Earth. The virus just doesn't have the biological key to infect it. This safely unlocks incredibly robust industrial production systems capable of manufacturing novel drugs and chemicals nature never even dreamed of.
Which brings us to our final, ultimate realization. We are successfully blurring the lines between what's natural and what's synthetically possible, right here in our own laboratories. So we really have to look up at the stars and wonder, if we can engineer alternative genetic codes, alternative elements, and alternative energy sources in just a few short decades, hasn't the vast 13 billion-year-old cosmos already beaten us to the punch? The alien life we're searching for might be out there right now, thriving on mechanics and methane, completely beyond life as we know it. Thanks for joining me on this explainer, and keep questioning what you think is possible.